Hey biology students, Professor Gideon here. In this lecture video, we're going to cover the adaptive immune response of the third line of defense, specifically the cell-mediated response. So the third line of defense, we actually break it into two categories. So the cell-mediated response is the response that involves T cells. So it's the T cell response. So I thought I'd set up an infection situation that we can explore together and describe how the T cells respond in the third line of defense. So I'm going to show you a diagram that I made. And yes, the nose is huge and everything is not exactly in proportion. So just bear with me here. I'm trying to explain to you how viruses like coronavirus and other cold and flu viruses invade the body and how the T cells would respond to that infection. So recall, anytime there's an infection, first that pathogen has to get through the first two barriers of defense, the first and the second line of defense. And let's talk about droplet transmission real quick. So you've been hearing that term referred to how coronavirus is spread from person to person. And so what happens is you have to be in contact with somebody who's coughing, and when they're coughing, they would be spewing out little viral particles. And so this is a virus. We'll say this is a virus here, this little green circle with all these little spikes. Okay, and that virus, if somebody had been coughing nearby, would be in a droplet, would be in a mucus droplet, and they're saying within about 6 feet distance from somebody who's actively coughing with coronavirus, they will release these respiratory droplets that contain viral particles, and all you need to do is just breathe those in. So if we breathe those in, now the virus has to get past the first line of defense. So in the diagram, do you see that the viral particle is entering the nostril? So it's getting past the first line of defense, which would be those nose hairs. So these represent nose hairs here. And so the virus might get stuck, might just get stuck there and not progress. Also, another part of first line of defense would be, let's say it goes in through the mouth. In the back of the throat, we call that the pharynx. There's all these layers of mucus. So the viral particles could get stuck in these mucus layers. And then remember, if that mucus then, if that viral particle then goes into the esophagus, that is all part of first line of defense because the esophagus leads to the stomach where there's our nice internal bag of acid called the stomach that will destroy or denature the viral particles. So it is possible to breathe in viral particles. We actually do it every day, and those viral particles never do cause illness in you because your first line of defense is effective at blocking the virus from getting to its target cells. So it gets trapped in these physical or chemical anatomical barriers that we call the first line of defense. So to progress to disease, the virus would have to invade target cells. So I'm showing here that the virus will enter through the nasal cavity, and if it doesn't get stuck here in the nose hair, it will progress to the target cells lining your respiratory canals. So you get all these you have a sinus cavity in here, you have the back of your throat, you have all these cells back here. These are ciliated cells of the respiratory tract. And viruses like coronavirus and common cold and flu, they love these cells, and so they bind to these cells and they invade those cells. So that would be a breach of the first line of defense if they're able to get past mucus lining, the stomach acid, the nose hair, they're able to get past all of that. Well, their job, the virus wants to get into its target cell. So I want to show you what that looks like here. So I've enlarged a target cell that coronavirus would want to invade in your respiratory tract. And the way viruses work, they have evolved to bind to specific receptors that we have on our surface of the cells. These are just normal receptors that our body uses as a way to communicate with other cells. And there's all sorts of different types of receptors on the surface of cells. It turns out that the coronavirus binds to a receptor called the ACE2 receptor. So what happens is that the virus is able to stick to the ACE2 receptor. So it sticks to the receptor here, spiky virus, sticks to the receptor. And that actually is a way that viruses have evolved to essentially have the key into the house of the cell. So it's like if you had given your key to somebody who you wouldn't actually want to enter your house, a burglar, a robber, somebody you wouldn't want to enter your house, but somehow they got a key to your house. And so that's what the virus is. The virus is a pathogen. We don't want it to go into the cell, but a lot of these viruses have co-evolved with our cells so that they carry the key to enter the lock. And so basically the virus unlocks the cell by binding to the ACE2 receptor. And by the way, this is specific to coronavirus because all viruses have their particular target receptor and a particular target cell. So anyways, the virus, when it binds, it unlocks the cell. So that allows the viral particle to enter the cell, enter the cytoplasm of the cell. And then that virus ultimately initiates copying of itself. Now we'll study viral replication in more detail in a future unit. So I didn't want to go to all of the details about viral replication. So let's just keep it simple for now. So we get viral replication that occurs inside of the cell. And that means that the virus is using the cell's machinery to do that. So it's using the enzymes of the cell, it's using the ribosomes of the cell, and it's doing that because, recall, viruses are not cellular, so viruses don't have ribosomes and they don't have any DNA polymerase or those sorts of things. So they have to use the cell. A lot of times we use the term hijack a cell, that the virus hijacks a cell and turns the cell into a viral cloning factory. So now the cell has been hijacked by virus, and it's going to be told by the virus, basically by the genome of the virus, the nucleic acid of the virus, to copy new viral particles. And that's how the virus replicates itself. It cannot replicate unless it gets inside of its target cell and uses the target cell's machinery to do that, cellular machinery to do that. That's different than bacteria. Bacteria can survive on surfaces and can actually multiply outside of the cells. They don't need your cells to do that because bacteria themselves are cells and they contain all of the necessary components to replicate themselves and divide, as you know. But viruses, they need to get inside the cell, so that's really important to their life cycle. So they replicate. And we're not just talking a couple. So here I just showed a few viruses are being replicated inside the cell, but this can be on the order of 10,000 new viruses from one cell. So imagine this is somewhere around greater than 10,000 plus new viruses. New viruses are being synthesized or replicated inside of one of your cells, one of those cells in your respiratory tract. And then what happens now that you have 10,000 new viruses being born inside of your cell is that they fill the cytoplasm of the cell and then they rupture the cell. So they release out of this target cell. And as they are released out of the target cell, well, they rupture the cell. So this target cell, which was a cell in your respiratory tract, and actually specifically for coronavirus, the target cell is a type 2 pneumocyte, which basically is a respiratory cell. So these are cells that line your respiratory system. So cells of the respiratory tract. So as the viruses are released, this cell dies. So we say cell lysis, cell death. So terrible news for this cell because it gets turned into a viral cloning factory and then it bursts as new viral particles are released. And then those viral particles will go out and search for more type 2 pneumocytes and will invade those. And pretty soon, it doesn't take long for there to be exponential number of viruses replicating inside of your cells. So one thing that I think you should know here is that the cell, the target cell here, it actually will display viral antigens on its surface. It'll do that using an MHC1 protein. So in a previous video, we discussed that there are receptors called MHC molecules that move to the surface of cells and act as recognition markers. So there's class 1 and class 2. Now an infected body cell like this would have MHC class 1. Recall MHC class 1 is found on all nucleated body cells. And what it'll do is it will actually bind to some of the viral antigens. So this would represent this little green ball here. This represents a viral antigen. And then it will push that MHC1 complex to its surface. So now it's wearing basically a badge showing that it is infected with this viral antigen. This specific viral antigen is what has caused this invasion, and it'll wear that on the surface. So it'll actually be wearing that, and that's going to be important to understand how the T cell is going to recognize what cells of the body have become infected with virus. Also, this cell that has died will be releasing inflammatory cytokines. So we'll show that as well. So this cell is now going to be dead. Okay, so dead or dying as it is lysed. And so it's going to release some inflammatory cytokines because that's what injured cells do. That's what injured cells or dying cells do. They release inflammatory cytokines. And then those inflammatory cytokines are signals, recall, and they begin the inflammation process. So let's go back to here. So here we have that the first line of defense was breached. The virus was able to reach its target cell. It's now replicating or breeding inside of target cells and releasing thousands of copies of itself that now go and infect more cells, and this is just getting exponential. So inflammatory signals are now being sent, and those inflammatory signals are going to initiate inflammation. And so let's show some of those inflammatory signals coming out here. And those inflammatory signals allow for inflammation to be initiated. Here I want to show you a lymphatic vessel. So a lymphatic vessel now is vasodilating. So we're getting vasodilation in this lymphatic vessel, which is why I showed it as a dotted line rather than a solid line, because we've learned that vasodilation means that it becomes leaky. So gaps form in between the cells that line the vessels, and that's so fluid can leak out, but specifically white blood cells can easily move out of the either lymphatic vessel or blood vessel, and can now go and attack. So this vessel now becomes leaky. So fluid is leaving the lymphatic vessels. So we get fluid leaking out, fluid going here. Well, what is one of the initial signs that you are getting a cold? A runny nose, right? So let's talk about the symptoms of cold flu viruses, and along with that also coronaviruses. So one of the initial symptoms would be a runny nose. Why do you get a runny nose? Well, it's because vasodilation has been initiated, and really it's these lymphatic vessels. If you recall, lymphatic vessels are filled with a clear fluid called lymph. So as the lymphatic vessels leak, that fluid goes into your sinus cavity, and then it starts coming out your nose, and so you start getting these droplets of fluid. This would be the runny nose. Also, if the virus has had a chance to make its way farther down from your sinus cavity through the back of the throat, it starts getting down here. Now, this is not perfect to, this is not anatomically perfect in my diagram at all, but this is supposed to be the trachea. So this would be the windpipe here. We've talked about it in previous lectures, but recall it has all these little ciliated hairs. So this is also a target cell for our little virus. So it also wants to bind there to the ciliated cells in the back of the throat. Well, recall now what's going to happen. White blood cells are coming to the scene because they've responded to the cytokine release. So vasodilation is occurring in lymphatic vessels, fluid loss is occurring, and then we're also getting these white blood cells that are coming to the scene, and they are squeezing out of the tissues. So we get these white blood cells showing up, and these white blood cells will squeeze out of the tissues. And let's say this white blood cell is going to come down here and try to eat this virus. And remember, these are phagocytes that initiate the second line of defense, because now we're in second line of defense. Now that we have inflammation, now that we have the leukocytes, we have the phagocytes, likely neutrophils or macrophages or dendritic cells coming to the scene of the infection, we're still in second line of defense. And so these phagocytes come and they try to eat the virus. Now, what about another type of phagocyte called a natural killer cell? Remember those? So the NK cells. NK cells release perforins, these toxic proteins, and they do that in response to the cytokine release. So they come to the scene and they squeeze out of blood vessels, or either blood vessels or lymphatic vessels. They squeeze out of the vessels and they go to the target cell, and then they just degranulate and release a whole bunch of perforin. What's that going to do? Well, it's going to destroy all the cells or a good number of the cells that are infected with virus. And so what happens is the cilia that line your trachea get damaged. And what happens then? Well, now it becomes difficult to clear the back of your throat. There's a lot of debris of all the cell death occurring along the trachea. So one of the other symptoms you get is a cough. So the reason why you cough is because those ciliated cells of your respiratory tract are damaged. Now they're also damaged due to the virus infecting them and destroying them and lysing them, but they're also damaged as a result of white blood cells like natural killer cells that are releasing perforins in the attack and destroying all of these ciliated cells. And the only way to clear all that debris out of your respiratory system would be to cough. And so that's why you develop a cough. Now, more as the signaling increases of cytokine signaling, we might have a fever response. In more severe cases, we do see a fever response. Not usually when you have a mild cold, but if you have the flu or if you have COVID-19, fever does seem to be a symptom. So that means that that cytokine response is just escalating, and then that message is being sent to the brain, hey, we're under attack, and the brain raises body temperature in response. So that's all part of second line of defense. And now you understand where those symptoms come from. So we don't want that virus to continue its path down the trachea because the next symptom that shows up for people who have coronavirus in the more severe coronavirus infection would be shortness of breath. So that means actually, before we go into that, that actually means that it's possible because people are recovering from coronavirus. Actually, a good percentage of people, the majority of people are recovering from coronavirus without death, without serious complications. What does that mean? It means that your second line of defense is probably doing the job. So those natural killer cells, they're killing out, they're wiping out cells in your trachea, they're taking out the virus, and that might just be enough. That might just be enough to eliminate the viral particles from further or continued infection. So that could be the case. However, if this is not enough, so that is where third line of defense comes in. If this is not efficient in eliminating the pathogen, then the immune system has to recruit third line of defense players. So we don't want that virus to continue down into the trachea. Please don't go down there, virus. Don't do that. Because if you do, where's that going to lead? Well, it's going to lead to the lungs. And we've talked about the lungs in a previous video, how vulnerable they are and how there's alveolar cells that can become invaded by virus. And that can affect oxygen uptake. And then that's when we get the shortness of breath symptom related to coronavirus. So we get the shortness of breath because the alveoli are infected and rupturing, and gas exchange is very difficult. And that is an initial symptom that we're talking about, somebody who has the viral pneumonia complication. And then things start to get serious there. So we need backups. We need backup. We need backup from the third line of defense. And really, that involves T cells and it also involves B cells. But in this video, we're going to just talk about the T cell response. So in a previous video, we described the process of phagocytosis. So I want to revisit that concept with you. I already did this diagram because we have drawn it out step by step together previously, but let's go over it. So here we're talking about this spiky green virus. This would be coronavirus. So the macrophage engulfs it in a phagosome. Remember, there are then these digestive vesicles called lysosomes present in macrophages that allow the lysosome to fuse with the phagosome. When we get the fusion, we get something called a phagolysosome. Then we get digestion of the virus. We can get release of some of those viral particles, although many of those viral particles, and remember we call those now antigens, so this would be a viral antigen. So the viral antigen is complexing to the MHC proteins, the MHC class II proteins, and then that's forming a complex that gets pushed to the surface of this cell. And when that happens, we now say that this macrophage is an antigen-presenting cell. So we call this macrophage an antigen-presenting cell because it is wearing the viral antigen complex to its MHC2 protein. So now I want to add just one extra piece of detail to this diagram that we haven't talked about. Now recall that the macrophage can display MHC class II. However, it can also display antigen complexed to MHC class I as well. So I'm going to add that little detail to our diagram. So we have a class I MHC and a class II. So now let's add the viral antigen in green complex to MHC1, and let's push that MHC1 complex to the surface. So this is MHC1, and over here this was MHC2 complex to the viral antigen. So that's important to note because this antigen-presenting cell has the capability to present to two different types of T lymphocytes. So now imagine that the antigen-presenting macrophage migrates away from the site of infection through a lymphatic vessel and enters a lymph node. So this is a lymph node. And these lymph nodes are located throughout the body. And in the lymph node, the APC macrophage, so here comes the APC macrophage, enters the lymph node and encounters millions of different types of T lymphocytes. So I tried to illustrate that here by showing you we have T lymphocytes with various types of TCR receptors. So we have some T cells that are displaying a green TCR. So this is a TCR. We have some T cells displaying a red TCR, and we have some T cells displaying a blue TCR. And the color coding in this diagram is just to show you that the TCR has to match a specific antigen presented by the antigen-presenting cell. So we have actually millions of different lymphocytes that exist in our lymph nodes and in various other lymphatic organs as well. And that's because they are a result of a complex process of genetic recombination that occurs in our cells, in those stem cells, and produces all of these different variants of T lymphocytes that are all displaying a slightly different version of the TCR. And that's so that there will be one specialized type of lymphocyte that recognizes the specific antigen involved in this particular immune attack. So if you recall, the color that we were going with for our viral antigen was green. So therefore, as the antigen-presenting macrophage enters the lymph node, it's going to recognize the T lymphocyte displaying the green TCR. So I've actually shown that there are two copies here, two different types of T cells in my diagram, both displaying the green TCR. And that's because there's two different types of T cells that the APC macrophage presents to. And recall, the way we can tell the difference is we look at the co-receptor. So the co-receptor of this specific TCR is CD4. So we refer to it as CD4 positive. Do you remember what type of T cell that is? That's right, it's a helper T cell. So we write that as a TH cell. And then up here, we have another type of lymphocyte, T lymphocyte, with a CD8 co-receptor. What type of T cell displays a CD8 co-receptor? That's right, this would be a cytotoxic T cell or a killer T cell. So these two different types of T cells. So now let's draw in our antigen presenting macrophage. So it looks something like this. So here comes the antigen presenting macrophage. And remember, the antigen presenting macrophage is presenting antigen complex to its MHC molecules. So here's where there's a little bit of detail to understand. So the MHC1 complex to antigen will present to the cytotoxic T cells. So cytotoxic T cells only recognize antigen complex to MHC1, and the helper T cell recognizes MHC2 complex to antigen. So remember, we have these green viral antigens. I'm going to circle that. So we have these green viral antigens complex to MHC, and now that's being presented to a cytotoxic T cell and to a helper T cell. And when we get that very specific binding interaction between these complexes, then we also get activation with interleukins. So there's an interleukin signal that would be sent from the antigen presenting macrophage to the helper T cell. That's going to activate the helper T cell. And then we also get a signal from that antigen presenting macrophage to the cytotoxic T cell. And we also get an interleukin signal from the helper T cell to the cytotoxic T cell. So this is the activation of a helper T cell and a cytotoxic T cell. And what's going to happen next is they're going to divide. Okay, so the TC cell and the TH cell, we call this process, by the way, we call this clonal selection. We've selected here for the specific T cells that we need to fight this infection. Notice we also have other lymphocytes in the lymph node, but they are not called into action for this particular attack because the TCRs on this lymphocyte and this lymphocyte do not recognize or bind specifically to the particular antigen in this infection. I always found this process analogous to the cartoon book that I remember playing as a kid, the Where's Waldo cartoon. So in the Where's Waldo cartoon, you have to search through these very elaborate pictures of
people, and you have to locate Waldo. So you could see here, Waldo's in the middle. And this reminds me to what happens inside of the lymph nodes when the antigen-presenting cell enters the lymph nodes and has to search for the specific lymphocytes that match the antigen involved in this infection, because there's millions of different lymphocytes in there, each unique in the TCR, and it has to get in there and search to find the correct one. So once that particular cytotoxic T cell and helper T cell are located, we get the clonal selection process. And what that means is that first we get selection of the correct lymphocyte, and then the lymphocytes, the TC and the TH lymphocytes, will clone themselves. Basically, they're going to go through a very rapid mitosis process to make millions of copies of themselves. You can actually notice this in your own body when your lymph nodes start to swell. So one of the signs of clonal selection would be swollen lymph nodes. A lot of times you can feel that around your tonsil area, in the neck. And I always find in myself when I'm sick, that is a good sign, actually. It's a sign that Waldo has been found by your antigen presenting cells, and they've located the lymphocytes that we need to fight this infection. And the lymphocytes are responding to the interleukin signaling and cloning themselves and making millions of identical copies that will then now exit the lymph node and go to the site of the infection.
Now back to this
Picture.
So the CD8 positive T cells get stimulated by helper T cells. And by the way, those CD8 positive T cells will also be stimulated to divide. So they will also start dividing. And they go to the scene. And when they go to the scene, they bind to cells, target cells, body cells that are displaying antigen complex to MHC1. So wherever they find a specific antigen, now so this represents a cytotoxic T cell. And recall, cytotoxic T cells have a CD8, so they have this CD8 co-receptor, and then they have a T cell receptor. And this T cell receptor I'm trying to show is green because it's trying to recognize the green antigen, and that's just trying to show you that there's a lock and key interaction going on, that really important theme in biology, that shape matters, shape is key when it comes to how molecules interact with each other. So this TCR of this particular cytotoxic T cell will recognize the antigen, and so it binds to this virally infected cell, and then what it does is it starts secreting perforins. So it starts secreting perforins. Perforins are toxic proteins. They're also secreted by natural killer cells. The difference, though, between a cytotoxic T cell and a natural killer cell is that cytotoxic T cells have TCRs, which means they can specifically come in and bind to cells that are virally infected. They know exactly who they're looking for. And here's another analogy. It's a lot like hound dogs. So in a lot of times missing person cases, they use hound dogs, and maybe you have a sock or a shirt of the person you're trying to find, and you have the hound dog sniff the scent of this missing person from some article of clothing, and then the hound dog has a whiff of the scent, and then you send that dog out into the woods looking for the missing person, and the hound dog can trace the scent to exactly where that person is. And that's what the cytotoxic T cells have going for them. They are, remember, this is specific immunity. So they can recognize a specific antigen where natural killer cells can only respond to interleukin response, in which case they degranulate in a non-specific manner. But cytotoxic T cells, they're like our hound dogs that sniff the scent of that particular antigen. Where is the virus replicating? Where specifically is it replicating? Inside the cell. Okay, get in there, kill the cell. So these perforins are actually going to kill the cell. Now, why do we want to kill the cell? Because this is our cell. Well, it's because it's breeding new virus. New viral particles are replicating inside. So you want to stop the replication. So the cytotoxic T cells are trying to locate any cell that has become hijacked and kill it before new virus can be packaged and synthesized and released. So ideally, you kill the cells before the virus is releasing out of the cell. You kill it while the virus is in the process of replicating, and then we take it out and we prevent new viral particles from being born and circulating. And that is what cytotoxic T cells do. They can only recognize antigen complexed to MHC proteins. Okay? So they can only recognize antigen complex to MHC proteins. And they go out, they seek out an infected cell, and they lyse that cell. I also have an animation of that process. Let's take a look together at that animation. Okay, so I'll walk you through it here. So now they're showing the virus infecting a target cell, like what I was trying to draw for you, that the virus gets inside of the cell, but some of the viral antigens get complex to MHC1. And this is, again, of the target cell. So the MHC1 is complex to viral antigen. Okay, here comes the cytotoxic T cell that recognizes that particular antigen because it has the matching TCR. So it recognizes the antigen complex to MHC1. It releases, here they're calling them cytotoxins, but those are usually perforants. And then it releases the cytotoxins to kill the cell that's infected. But it's also important to understand that this process would not occur without signaling from a helper T cell and without that antigen presenting cell locating a helper T cell and then activating that helper T cell, which then in turn activates the cytotoxic T cell. Here's a picture showing, here's a picture showing that process of activating helper T cells. So it's showing the antigen presentation process within a macrophage. It's showing that the macrophage is binding specifically to a helper T cell, activating that helper T cell with interleukin release. And then this is the microscopic view of that happening. So this cell on the left is a macrophage. This cell on the right is a helper T cell. And this is how we're activating that helper T cell. So let's summarize. Let's summarize what is called the cell-mediated response. So what happens is there's a pathogen. The pathogen gets engulfed by an antigen-presenting cell. That antigen-presenting cell activates a helper T cell, and then that helper T cell activates a cytotoxic T cell, and that pathogen is invading target body cells. And this is specifically true of all viruses, so all viruses, this is what they're doing, but also some bacteria and other types of pathogens as well. But not all bacteria invade inside of our body cells. So, but all viruses do. This is how you have to eliminate a viral infection. So now we have an activated cytotoxic T cell, and then that cytotoxic T cell will come and it will bind to the MHC. So here we have an MHC1 antigen complex on that targeted body cell, and the TC cell will recognize that, and then we get cell lysis. And the purpose of that is to prevent pathogen replication inside of the cell. And then some of these cytotoxic T cells will differentiate into memory T cells. So memory T cells, we have far more copies of these memory T cells, and they are going to patrol the body, and in the event that that same virus invades, as long as it has the same antigen, the virus hasn't mutated and changed its antigen components, but if it has the same antigen, the memory T cells will recognize that pathogen and eliminate that pathogen faster than normal, because we won't have to go through all of this antigen presentation and activation of helper T cells. It'll just go right and start killing any cells, any body cells that are displaying MHC1 complex. Okay, so in the next video, we'll talk about the B cells, the B lymphocytes, because that is the second branch of the response.